Good morning, everyone. Thank you for being here. We're going to start off with the case of the week by Sam Reinhart, and after that, Dr. Cohen will introduce Dr. Loscalzo.
Okay, our case of the week is a 72-year-old man who presented to the Advanced Heart Failure Clinic or was referred there with shortness of breath and dyspnea on exertion. He has an extensive cardiovascular history, including coronary artery disease with 2 prior PCIs and a known chronic total occlusion of the RCA, heart failure with preserved ejection fraction, moderate to severe MR, recurrent ventricular tachycardia with an ICD and an ablation, as well as on chronic amiodarone, paroxysmal atrial fibrillation on apixaban, COPD on chronic oxygen, CKD stage 3 to 4. He's had an abdominal aortic aneurysm stent repair, and then bilateral carotid endarterectomies. His fluid management as an outpatient proved quite difficult, given his chronic kidney disease and his mitral regurgitation. So decision was made to place a CardioMEMS device to assist with his outpatient fluid management. His right heart catheterization in August during CardioMEMS placement showed the following pressures with an adequate cardiac output and index. Over the preceding months, his diuresis with assistance from his CardioMEMS allowed for him to be weaned off oxygen at rest, and his lower extremity edema improved significantly. However, he remained significantly dyspneic with exertion. So his exam at that time at reevaluation showed a normal vital signs, and again, you notice he's saturating normally on room air. He was in sinus with regular rate and rhythm, but a holosystolic MR murmur at the apex, mild crackles at the bases on lung exam, and just trace edema now significantly improved. His transesophageal echocardiogram showed low normal systolic function with some wall motion abnormalities, normal RV size with mildly decreased function, and moderate to severe posteriorly directed mitral regurgitation, primarily thought to come from the P2 and P3 leaflets. So because his resting hemodynamics had significantly improved, but his dyspnea on exertion had not, the decision was made to proceed with MitraClip. Here we have his left atrial tracings from the procedure. His mean pressure in the left atrium was 24 millimeters of mercury with V waves up to 50 prior to the device deployment. And then post, his mean pressure decreased significantly to 8 millimeters of mercury with V waves only up to 14. So interestingly, for this patient, now that we have a CardioMEMS, we're able to monitor his pressures. And his echo post-procedure showed he had just mild intravalvular regurgitation with no stenosis associated with the device. So his CardioMEMS readings, the blue arrow up there denotes his day of hospital discharge. It was just last week, so we only have a few readings. And again, these are at rest. So we haven't seen a significant change as of yet, but we're hoping that given that his MR seems to be affecting him more with exertion, that we'll see significant improvement with the device, and we may see his diuretic requirement come down as well. So that's all I have.
How do you introduce an icon? The answer to that is with brevity. Because if I were to start to enumerate all the achievements, honors, and awards that Joe Loscalzo has had, I would cut very significantly into his lecture. So I'm not going to do that. Joe Loscalzo is the Hersey Professor of the Theory and Practice of Medicine, which is the same chair at Harvard Medical School and the Brigham Hospital that was held by his predecessor, Gene Braunwald. He is also the Soma Weiss Distinguished Chair in Medicine at Harvard, which in itself and in its history is quite remarkable. Soma Weiss, just as a sidebar, was born in Transylvania, which is now Hungary. and he's born in 1898. He came to the United States at the end of World War I. After a year or so, he entered Cornell Medical School and was graduated. And over the next decade, came to Harvard, where at the age of 39, he became chair of medicine. He had a brilliant career. He was the first person to describe the carotid sinus hypersensitivity syndrome, and in conjunction with G. Kenneth Mallory, he described the Mallory-Weiss syndrome, hematemesis after retching extensively. At the age of 43, he developed a severe headache, is said to have walked to the emergency room at the Brigham, said, "I've had an intracerebral bleed." He dropped into coma and died 2 days later. Now back to Joe Loscalzo. I will just state that he has authored or co-authored over 1,000 scientific manuscripts and edited, authored, or co-edited 50 books and holds 32 patents. It is particularly fitting today that he speaks in large part through the generosity of the Calabrese family, with whose support these grand rounds are given each year. It's also more than worthwhile to state that what goes around comes around, because about 3 decades ago, Guido Calabrese, who was dean at Yale Law School, and his brother Paul Calabrese, who became chair of medicine at Brown, developed or funded a lectureship to honor Massimo Calabrese, their father. Massimo Calabrese was a cardiologist in Italy in the 1930s, and as fascism grew in Italy and Benito Mussolini persecuted people, Massimo in about 1937, 1938 took his family and came to the United States. Massimo got an MPH at Yale School of Public Health, and in the 1950s and 60s, he was at our VA hospital, and he was perhaps our only faculty member at the VA hospital. His specialty was electrocardiography, and he taught generations of Yale medical students how to read electrocardiograms. The Calabrese family, as I indicated, Guido Calabrese, Dean's brother Paul, funded the Massimo Calabrese lectureship in the late 1980s, and the first such lecture was in 1990, 3 decades ago. The very first lecturer in 1990 was Joe Loscalzo. Join me in welcoming Joe Loscalzo back to Yale. Joe, welcome.
Well, thank you very much, Larry, for that very kind introduction. And thank you all for being here today and for welcoming me so warmly for this visit. I had a very enjoyable dinner last night with lots of old friends and former trainees and colleagues. And it meant a lot because it really helped me think in stark reveal about how many people I know here and how many lives we've touched together. So I thank you for the opportunity to be here. So I'm going to talk about something a little different from what you typically hear at a cardiology grand rounds, but I want to do so because I think that from my perspective, what I'm going to discuss is a strategy for redefining human disease in general and for approaching therapeutics in a more rational way in general, but with a cardiovascular focus. So let's begin with 2 key questions. The first is, why has the rapidly expanding knowledge of the human genome not yet given us the expected epiphanies about complex human disease that's been so deeply promised? And the second, why has new drug development been limited despite a remarkable range of technologies that now can be used to identify drug targets and design suitable candidate drugs? And the fact that I raised both questions on the same slide suggests, or should, that the answer will be similar for both. So let me give an example of what the problem is as I see it. And it's simply that we try to render very complex biology into something that's much too simple. So here let's begin with this, the homocysteine theory of atherothrombosis, with which you're all familiar. This was first proposed by Kilmer McCulley in the late 1960s after conducting autopsies on young individuals who died with homocystinuria at the MGH. And he proposed that mild degrees of hyperhomocystinemia might be a risk factor for atherosclerosis. And over the next 30 years, multiple epidemiological studies provided evidence for this hypothesis, so much so that clinical trials began in the late 1990s. And to frame the issue, as you can see in the last bullet point here, between 20 and 40% of patients with conventional vascular disease have hyperhomocystinemia of a mild to moderate degree, but only 2% of the general population does. So those clinical trials were predicated on the importance of B vitamins in homocysteine metabolism, and I highlight that metabolism here. For transsulfuration through to sulfate, one needs B6, and for remethylation to methionine, one needs vitamin B12 and folate or betaine as an alternative. So based on that simple biochemistry, 7 clinical trials began in the late 1990s, the first 2 of which were published in 2006, I think in the same issue of the New England Journal, the Norvit trial and the HOPE-2 trial. And they had different designs, but the outcome was essentially the same, as was the outcome of all of the other trials published subsequently. That if one used combinations of B vitamins, felt to demonstrate a reduction in circulating homocysteine effectively compared to placebo, there was no effect on secondary events. These were secondary prevention trials or their equivalent in the case of HOPE-2. So following the null outcome of these trials, the clinical trial community working in this area began to pull their hair and gnash their teeth. How could they have been so wrong? Here we have homocysteine, which can easily be reduced in circulating blood by either remethylating it to methionine or transsulfurating it to cystathionine. What could be simpler? And in each of the trials, there was demonstrable evidence that homocysteine levels were reduced. Why were the outcomes not improved? But another way to frame this issue is to think about it from a much more integrative and holistic context. Here we have homocysteine metabolism linked to folate, inserted in folate metabolism, which has many other aspects to it in human intermediary metabolism, which of course sits in a small northwest corner of the intermediary metabolism map, as you remember from basic biochemistry. So the real question should be not why did these trials fail, but how could a rational person have considered the fact that they could have succeeded? And in fact, subsequently, or around the same time as the first trials were coming out, there was ample evidence that the use of folate and B12 through alternate actions, such as stimulating cell proliferation, could actually worsen outcome following, in this case, angioplasty. Homocysteine is also crucial for methylation and its consequent epigenetic effects, where the SAM-SAH ratio regulates methylation functions in virtually every reaction requiring methylation in the body. So this is a very complex metabolism that was vastly oversimplified. At the end of 2017, my colleague, and some of you in the audience remember Jeremy Green, who trained at the Brigham, is an internist, but also a medical historian and is now a professor at Johns Hopkins. He and I wrote a piece on the long arc of reductionism in medicine. And on the left, if you can see this insert clearly enough, it highlights this notion of reductionism, progressive reductionism, in this case, the approach to tuberculosis. In the 18th century, one referred to the sick person with TB, it wasn't called TB at the time, as phthisis. In the early 19th century, There were organ and tissue analyses performed using, at that time, histopathologic approaches, and caseating granulometer were first defined. By the late 19th century, one could identify the bacterium MTB, and then ultimately in the 20th century, lesions were analyzed at the molecular level. But now, of course, we're moving back toward a more holistic approach to tuberculosis, since many other features and factors contribute to overt manifestations of the disease as compared to containment of the disease by an adequate immune system, as an aspect of TB ecology with which we're all familiar. So that's one feature of this arc of reductionism, which has been certainly clearly beneficial in our dissection of disease up to a point. On the right, we have a picture of Paul Ehrlich. I'll come back to him a little bit later. And he really is the father of chemical biology, if you will. His notion of one disease, one gene, that term wasn't used at the time, one protein or target, and one drug, really set the stage for current aspects of therapeutics, drug development, organizations of departments of medicine, NIH funding mechanisms. These all stem from this notion that there's a magic bullet for every disease, no matter how complicated. But all of that being said, we sort of reached the limits of reductionism, in my view, at least in biomedicine. And the scientific paradigm for our approach to disease is therefore changing. Most biological systems are much more complicated. They respond to multiple inputs that vary simultaneously and can interact. These are complex systems by definition, and they form, in the cases I'll discuss today, molecular networks. And new approaches, new quantitative approaches have been developing over the last 20 years that now can be used to explore how these systems evolve, respond to perturbations, which could be disease-inducing perturbations, or respond to therapies as another form of perturbation. Now, what do we need to know about networks to understand the points I want to illustrate for disease for you? I'll just have two slides to highlight, or three slides that highlight features of biological networks to keep in mind. One is these are not random associations of proteins in complex networks. These networks are actually clustered, as I'll show you an example of in a moment, or scale-free, and I'll explain what that means in a moment. They manifest emergent behavior, which means that you can't predict how the network will behave by knowing everything you can about one element in the network. It would be akin to arguing that an electrical engineer could predict exactly how a circuit will behave, knowing everything he possibly can about the behavior of one resistor in the circuit. Now, you may chuckle at that analogy, but that's what we as biologists have been doing for the last 60 years, right? We focus on our favorite transcription factor, or our favorite protein, or our favorite metabolite. So understanding these emergent behaviors is really critical to understanding how a system will respond to a perturbation that could lead to a disease, or how a system might respond to a therapy or intervention. These modules are, the structural modularity of these networks also link to function. So they're modules that are contained within different compartments of the cell that are important to dissect if you're, for example, pursuing effects in the mitochondrion or in the ER. The clustered nature of these networks also facilitate the flow of molecular information across the network. It's much like cliques in a high school. You know, there are, I'm probably dating myself here using these terms, but there are jocks and there are nerds, and you know, usually once in a while you find a nerdy jock or a jockey nerd, and that person provides the weak link between those two cliques, and the flow of information, of gossip across the high school class is a consequence of that weak link. Same thing's true for molecular networks. There are weak links that link the clusters and facilitate the information flow. And there is, just like with the genetic code, there are canonical structural and dynamic motifs that reflect organizing principles in this systems biology that we're learning more about as time goes on. So here are two extreme examples of network structures. On the left, we have a random network, and it's described by a Poisson distribution. On the right, we have what's typically seen in biological systems, a scale-free network. And here, the distribution is log linear, with the slope being unique to the network and invariant. This is the notion of scale-freeness. And what that means is that as the network grows, these blue nodes, which are the hubs and the most highly connected elements in the network, accrue or agglomerate additional nodes with the growth of the network. And that's equivalent to Pareto's rule, the economist who pointed out that the rich get richer. That's true here as scale-free networks evolve. So that slope is invariant with respect to size and is a basic organizing principle of any biological system. Now, why do we need to know this stuff? What's the point of understanding a little bit of math and network science here? Well, there are many reasons to understand it, but two real implications of these scale-free networks for us as biologists and physicians is that first, they recapitulate natural selection and evolution. They define the difference between mutable nodes, which are weakly connected, the black nodes on the right here linked to the hubs, that are weakly connected. These engender diversity and facilitate natural selection. And immutable nodes, which are the hubs, the loss of which is lethal for an organism, such as G6PD, which can't be knocked out completely. It is embryonically lethal. In addition, scale freeness allows a network to accommodate perturbations, environmental perturbations, with minimal effect on the functions of the network or the organism, unless a hub is directly affected. And since there are so few hubs, the likelihood from a stochastic perspective that the network will go down with a random perturbation is far less than is the case for a random network. So there are real features of network structures, scale-free network structures, that in many respects accommodate and account for the evolution of biological systems, and as I'll try to argue, also the development of disease. So now let's turn to the way we think about complex systems in modern genomics so that I can highlight the problems with that approach. Here we have a Manhattan plot, commonly used to show genome-wide association of sites along the genome to a particular phenotype, in this case Crohn's disease. This plot was taken from one of the first large GWAS studies, Wellcome Trust Case Control Consortium. There were 7 diseases, as I recall, that were analyzed by GWAS in this paper. And as you can see, the green dots illustrate those loci along the genome that link at a highly statistically significant level to the presence of Crohn's disease. But the question is, what does that mean? And what do we do with these data? What we typically do with these data is we look at every one of the loci, try to understand its biology. If we can understand the biology, we decide whether or not that particular locus or the protein that's synthesized at that site in that allele might be a drug target for the disease of interest. Most of the time, we're wrong. PCSK9 comes along very infrequently. I mean, this is a problem with drug development as it stands because every pharma company is looking for the next PCSK9. They're not going to find one. This was an uncommon single association that was observed actually by looking at rare phenotypes in patients that had significantly elevated activity versus significantly reduced activity. So we really have to understand how these sites interact, how they conspire to create the phenotype to be much more precise and definitive about the pathogenesis of this and other diseases. Let me put it another way using another simple-minded analogy for my sake, not for yours. So let's say, for the sake of argument, that you are a new mechanic who's just begun his job in an auto repair shop, and you walk in one day and your boss has laid out for you all the engine parts for 2 cars. The cars have identical engines. On the left, all the parts are laid out for the normal functioning engine. On the right, there's a malfunctioning engine. And the boss says to you, you've never seen the engine assembled, you don't know how to put the pieces back together, but he says, what do you think the problem is? Or what do you think the problems are here? Why is the engine on the right not functioning and the one on the left is functioning normally? And you're a keen observer and you find these little chips, as I've highlighted in red, in gears. And you ask the question, well, perhaps one of these gears or all of them is responsible for the engine not functioning properly. But you don't really know that. These could be simple manufacturing errors which have no functional consequence. And in fact, many of the variants of uncertain significance in our genomic analyses wind up being just this. These are variants which have no real consequence to function. The only way to know whether they have functional importance is to know the assembly diagram. And the question is, how do you assemble the sites along that genome that you think are responsible for a disease in a way that is insightful and can give you useful information about the pathogenesis of the disease? Let me give you some evidence for the importance of this context, this network context, I'll call it genomic context, in understanding disease from a couple of interesting recent studies. So here, from Steve Friend's group in Seattle, there was an analysis of almost 590,000 genomes of individuals who many of whom had severe Mendelian childhood diseases. And when they analyzed these genomes, this group found that there was 874 genes screened comprehensively, that is, the whole exome was sequenced. They found in this analysis 13 adults with 8 mutations that should have led to severe Mendelian phenotypes. Cystic fibrosis, Smith-Lemli-Opitz, which is a dehydrocholesterol reductase mutation, EBS, Pfeiffer syndrome. All of these diseases, certainly the last 3, are by definition 100% penetrant, being classic Mendelian disorders. But in fact, these 13 adults had no evidence for disease. And what they concluded was these, quote, individuals are buffering the effects of rare, highly penetrant, deleterious mutations. Well, that's true. That's sort of a tautologous observation. The question is, how? What is it that allows these individuals to maintain normal physiology in the face of what otherwise would have been severe mutations? And I'll come back to the answer in a moment. Another important another paper that I think makes another important point about the role of genomic or network context is a paper by Raj Manrai, who was a graduate student with Zach Kohani and worked with our group as well. And in this paper, what he did was ask the question, are there differences by race that might account for misclassification of benign variants as pathogenic in patients believed to have HCM? And he made the observation first on the basis of the fact that HCM variants that were not variants of uncertain significance, but variants that were believed to be associated with a phenotype, were overrepresented in the general population. And you can see the pie chart on the left here. And when he looked at that overrepresentation by race, he found that many of these variants were much more frequent in African Americans than they were in white Americans, in whom there was a far lower likelihood of there being true HCM by conventional echo measures. And the sources for the genomes are listed below, all of which had largely Northern European whites as the basis for their analysis. And Raj made the case, of course, as others have in other contexts, that racial and ethnic backgrounds play an important role in allowing one to attribute or not a variant to a disease pathology or pathobiology. So context is everything. So can molecular networks define genomic context and give unique insight into disease pathogenesis? Well, I believe that the answer to that question is yes. And with my colleague Laszlo Barabasi, with whom I've worked now for over 12 or 13 years, Laszlo is a physicist who's arguably one of the top 3 network scientists in the world. This is a statistical physicist who thinks a lot about how to analyze complex networks in a variety of settings, from the internet to social networks to other physical networks. And his foray into biology began with some of the work he's done with us. And we published a concept piece in 2011 in which we set out this hypothesis that one can explore the universe of interacting proteins within which we hypothesize one could define modules or subnetworks that were specific to certain diseases, that the proteins and genes that code for those proteins cluster, just as I described clustering earlier, in very unique domains in this universe of interactions. And those can serve as the basis for analyzing mechanisms of disease and defining therapeutic targets much more effectively than we currently do. And we based this hypothesis on a few features. A simple analysis that we did on the left here shows that if you look at essential, these are hub genes, versus disease genes, disease genes in the Venn diagram have far fewer essential genes among their members than do non-disease genes. So chronic, complex adult diseases, I'm not talking about Mendelian disorders in newborns, chronic complex diseases of the sort we deal with tend to occur as a consequence of variation in genes that are also responsible for diversity. These are sort of failed effects or failed attempts by nature to adapt to an evolving environment. These are the flaws or consequences of those kinds of random efforts. On the right, you can see in this case, at this particular point, our cartoon version of the Interactome. And the kind of Interactome that we focus on is the network of interactions among proteins in a cell. So these are true physical interactions between proteins. And as you can imagine, given how complex the cell is, there are many different interactions that can be teased out. And we have to thank Mark Vidal, who's a biochemist at our place, who has been working using a variety of methods, yeast-2 hybrid screens and other physical-chemical methods, has been working on developing the true physical Interactome for several years now. We used his initial version of the Interactome to build our broader version of it. Why do we focus on protein-protein interactions? There are other ways to build molecular networks, but for today's purposes, we'll focus just on the protein-protein interactome. The reason that we do is because the genome implies phenotype, but the proteome is phenotype. It's much like the floor plan of a house shown on the left. It gives you an idea of what the house should look like, but you don't really know that, even with modern CAD technology, until you see it in front of you. And more important, as the proteome matures, as it's exposed to the elements, as it develops oxidative post-translational modifications, you can't in any way predict what that's going to look like based on the floor plan. So the proteome is a critical element here for analyzing sources of disease. Now we argued, again shown here schematically, that the Interactome, this universe of protein-protein interactions, and the version we have now accounts for probably about 30% of the possible interactions from our estimates, that within it there are domains that are color-coded here, that are specific to diseases. And our efforts in the next study, as I'll show you, were designed to identify those modules and begin to explore them on a disease-by-disease basis. So here's an illustration of a real Interactome, that fuzzball on the background. It's one of the earlier versions of the Interactome that we used. It had over 13,000 nodes or proteins and over 140,000 edges or links, which represent the association of proteins. This ratio is also interesting because you remember in basic biochemistry when your professor told you why proteins are as big as they are, the argument was that the active site or the binding site of the protein was so constrained that it required this massive infrastructure to hold it just where it needed to be held for function. But we know that's nonsense because you can make enzymatic catalysts out of far smaller molecules. What I think this ratio illustrates is that the reason proteins are as big as they are is because of all the interactions they have with other proteins. On average here, 10 to 1, different proteins. So I've mapped, again in schematic form against this real Interactome, the disease modules for 3 different diseases: multiple sclerosis, rheumatoid arthritis, and peroxisomal disorders. And notice that RA and MS have some overlap, as you might expect, given their autoimmune underpinnings. And we then spent considerable time analyzing about 199 different diseases, which had to have at least 20 genes that we could begin to construct modules through mapping to the Interactome with. And we found that, in fact, 85 to 88% of them comprised unique modules that were distinct. And they had very interesting features that time doesn't permit me to go into great detail about. But here's an example of 2 overlapping modules that illustrate a pathway that hadn't been recognized previously as common to these 2 diseases. And this is asthma and celiac disease. And the overlap in the module focuses on the immune network for IgA production. It makes sense that it should be important in these diseases, but it hadn't been explored previously. So from a cardiovascular perspective, let me just, in 2 slides, walk you through the way we develop a disease module. This is work that was begun by Vicky Parekh, who was then a Sarnoff Fellow in the lab, and Steve Chan, who was a postdoc in the lab at the time. We identify the disease phenotype of interest. We ascertain the network components, which are the genes or proteins that have been associated with the disease, either through conventional experiments or through a unbiased genomic analysis. We then construct the disease network by mapping these components to the interactome and see whether or not that interactome is unique and circumscribed. Here's an example of the PAH disease module constructed in this way. You're not meant to read this in detail, but if you just stand back a little bit and notice a few features, you can see that we've separated it artificially into 8 different modules, but each of these modules has, as I mentioned earlier, modularity has some functional correlate. So if you look at the extreme right module, the yellow module, you'll notice that this is the PDE module and the guanylyl cyclase module. Makes sense that they should all be linked together. And similarly, other modules have similar kinds of functional schema. We updated this, Rusheng Wang did just last year. Now, what other information can this kind of network analysis give you that could be helpful in dissecting disease? Well, it can inform GWAS. Here's a great example of that. Jörg Menk, this was in the supplement to the paper, took type 2 diabetes, for which there have been about 200 loci linked. And these loci all had very tiny effect sizes, but they reached genome-wide significance, except for the first 15 or so. So what he did next was he mapped each of those loci to the interactome and followed the z-score, which is in the middle panel, for clustering. This is a statistical measure that gives insight into the likelihood that this clustering is greater than the play of chance. And as you could see, every so often the z-score would bump up considerably. And it also began at a very high level and stayed above 3 pretty much throughout the entire 200 loci. Now, down below you can see what gene was added. These were added ad seriatim based on their GWAS p-values in the upper graph. What gene product was added to allow for that increase in clustering. And here's an example of a pattern that highlights how the addition of a single gene, in this case GRB14 or COM2 or PRKC alpha, brought together a group of proteins that otherwise hadn't been linked. So these served as the missing links that tied together the association of different pathways that therefore gave novel insights into mechanisms that hadn't been appreciated previously for type 2 diabetes. Now, another way to use, so there are other examples that we published in that paper and other papers that I'll just summarize in a little bit for you. But I think these cases highlight the power of using a network strategy for dissecting molecular determinants of complex disease. Another way to think about systems pathobiology is not from the perspective of well-defined, classically defined Oslerian diseases, but to think about the endophenotypes that are associated with all diseases, such as inflammation or thrombosis or fibrosis or proliferation, apoptosis, necrosis. Are there distinct features or signatures that can map to the interactome for these endophenotypes? And does that give us insight into disease mechanism or treatment? So here's an example from another group, Wang at San Diego, in which they took 11 different disease models, these are all in rodents, from 9 different tissues and looked at gene expression patterns for those genes that had been associated with inflammatory responses. This was in 2012. And I can't remember the exact number, I think it was about 300 or so. And as you can see, the heat maps show pretty much a similar pattern for all these different diseases. The type 2 diabetes, a DBDB mouse, stroke in the TMCAO brain, obesity, sarcopenia. It's pretty striking how similar those are. And that raised the issue of whether or not we should begin to think about the endophenotype risk that we have that's defined on the basis of this kind of analysis and preempt its expression once a second insult occurs that might lead to an organ-specific problem, like hepatitis or acute myocardial infarction. Why do I say that? Well, let's step back for a second and look at what Dina Gassian did, who was a graduate student with us, did to explore these endophenotype modules. She was able to define, in this case, 3 different modules, one for inflammation, one for thrombosis, and one for fibrosis. And as you can see, there's a great deal of overlap between the thrombosis and inflammation module and between the fibrosis and inflammation module, with only a few proteins that are distinct from the inflammation module itself. And when you map these modules to the interactome, they sit squarely in the center. And they touch on about 60% of all of the disease modules that I mentioned earlier, arguing that these common endophenotypes may govern the extent to which disease is expressed in a particular organ or organ system when an insult occurs in that organ. And perhaps that might be another way to approach disease prevention by, at birth, determining whether or not you are excessively pro-inflammatory or excessively pro-fibrotic, and sorting out a definitive therapeutic way to guard against those adverse responses for future protection. The information about these endophenotypes has also given us insight into specific, unique disease targets. And this is work that Brad Marin and Semikin have done, recently published last year, in which they used a network analysis of fibrosis endophenotypes, which they categorized as two different types: pathologic fibrosis and reparative fibrosis. And straddling those two fibrotic modules is, as you can see highlighted in the right lower quadrant here in D, a specific scaffold protein called NED9, which is responsible for governing the regulation of the expression of collagen 3A gene expression. NED9 is itself critical in facilitating in patients with PAH perivascular fibrosis. And for that reason, it's become a target, a potential therapeutic target that Brad is trying to develop at this point in time. It would never have been recognized as important in this vascular disease without a network analysis. The only area in which NED9 had been studied previously happened to be in an adenocarcinoma cell model, where it turned out to be an important regulator of stromal synthesis. So without going further here, I just want to summarize for you some of the network-based discoveries of disease mediators that our group and other groups have published in the recent past. I talked about PAH and fibrosis. Sharma published a paper last year that focused on a novel transcriptional program that has been identified by a network analysis that governs type 2 diabetes. Sharma and colleagues in 2015, using an asthma module analysis, identified a pathway that's responsible for steroid-resistant asthma. A transcription factor they were able to tease out in the network seemed to actually be pro-inflammatory. It stimulated an inflammatory pathway in the presence of a corticosteroid, interestingly. Schlatter and Elena Aikawa's group explored the pathways that are responsible for valvular calcification, aortic valvular calcification, and found 3 novel pathways that hadn't been previously appreciated. And Mirzakhani with Scott Weiss have identified vitamin D regulatory pathways that overlap with the preeclampsia module and link specifically to his epidemiological studies that had shown vitamin D ingestion by the mother early in pregnancy limits the likelihood of developing preeclampsia later in pregnancy. Now, there are other kinds of networks that we could talk about. I'll just show you one other kind of network approach to disease, and this has to do with physiological phenotyping. We've talked about molecular networks. These are physiological networks, and this is a very nice study by Will Oldham and Brad Marin, published last year, in which they took over 700 patients who were undergoing ICPET for exercise intolerance and dyspnea. And for those of you who have done or know how to read ICPET studies, know that there are about 78, actually 78 precisely, different physiological parameters measured during an ICPET study, but only 2 of them are actually used clinically. So there's a lot more information there than has been mined previously, and this is what Will and Rusheng Wang did. They explored the links, the physiological associations among these parameters, eliminated the collinear parameters, and came up with a much sparser subnetwork, shown in B, of parameters that could predict very nicely all-cause hospitalization and all-cause 3-year hospitalization as a function of 4 different strata of that particular signature. So another way to put all this together is that we need to focus on what I would call network-based precision medicine to get to appropriate diagnostics and therapeutics, as I'll talk about in the last 10 minutes. And we know about genotypes and phenotypes. I'm adding between them now this notion of reticulotype, which is this is from the Latin for network. This is a network that is specific to an individual in which the genetic variants or the protein-coded variants that may exist in some cases to protect against a disease or in other cases to promote a disease interact and are associated with some variants in the endophenotype networks to create the phenotype. And it is only through this kind of linkage among network types that we can actually, I think, dissect disease rather precisely. And Jane Leopold and I made this case in a review piece in Circ Research last year, which highlights exactly the same points, but adds the notion that there are also other networks that are important to begin to explore, which we have not, including the microbiome, the metabolic network, the exposome, other features that might interact and conspire to create these complex phenotypes. So in the last 10 minutes, let's talk about drug discovery and how network analysis might help that process. Here you see up through 2016, FDA-approved drugs. These are new medical entities, new compositions of matter. And as you can see, it's been sort of sputtering along at around 40 or so for the last 10 to 12 years. There was a bit of a spike this past year, but then it's not clear what will happen this coming year, of course. And if you ask the question of drug developers why productivity has been declining, because I should point out in the decade before this, the numbers of approved drugs were more in the 50 to 60 range per year. They'll give you the usual answers. The regulatory environment is extreme. There's an increasing need to explore new targets. Easy targets have been exhausted. There's an increasing attrition rate for developing drugs, in part owing to regulation. But I would say that one of the major reasons for declining productivity is this flawed reductionist approach to drug development, the need to define or identify a magic bullet, again, thanks to Ehrlich. And remember that Ehrlich, this is not to trivialize what he did. He was really a striking scientist. His observations were derived from his notions, his observations on the tissue selectivity of dyes. And he noted that tissues stain differently, something we take for granted, and argued that there probably were different, as he called them, side chains that might account for the reactions to these dyes that allowed for this differential staining. And he then went on to expand this notion into one that we ultimately referred to as a receptor notion of disease against which a drug could be developed. He performed the first, if you will, low throughput screen, pulling drugs off the shelf. And of course, he found, as he humbly called it, Ehrlich's preparation 606, which was the arsenical that was used to treat malaria. And we were off and running with our current strategy for drug development. But here's a network view of drug development that I think highlights what some of the problems are. We have this schematic diagram here. Just look on the right of a disease module. And in red, you can see the pathways that are affected by the variants in that disease module in that person. And there's a drug target, which has been identified by conventional means. And that drug target has a function which can be assayed, so one can screen for the effects of drugs against that target. But the problem, of course, is that there are probably other targets, and I'll highlight how many others there are on average in a moment, that are not being screened for, some of which may, in fact, cause adverse effects that can only be appreciated after the drug's been released or approved, if it's not very dramatic or very frequent. And there may also be other drug targets which could be useful for other diseases that haven't been explored because that wasn't the development program that was used to identify this target. So we took that last point to heart and thought a little bit about how one might use network structures to repurpose drugs that have been approved. And let's put this another way. Here we have a disease module highlighted in light green here. And you can see the disease genes that are known are listed in red. The drug targets are listed in triangles, purple triangles. There happens to be, our hypothesis is that there's another target T2, which sits outside the disease module and may be close to the disease module of interest and could serve as an alternate target for which an existing drug could be repurposed. So this is work of Feixiang Chang and Rusheng Wang, who performed the following analysis. The plan was to identify disease genes in the PPI for a disease of interest, and we chose myocardial infarction, as I'll show you in a moment. Identify the FDA-approved drug targets in the PPI that sit within that disease module or near it. We then used a population-based strategy for validation, and I'll explain what that means in a moment. And then we went back to the experimental lab and confirmed the mechanism of action of the repurposed drug. So this is here you can see the schematic diagram of the cardiovascular, cardiovascular modules that cluster together in the interactome. And when we explored the FDA-approved drugs whose targets are near, if not within, the MI module, but not used for the treatment of MI, we found 4 that were of interest: carbamazepine, lithium, azathioprine, and hydroxychloroquine. And the question here is, what do we use as controls? Well, we need to use drugs that are used to treat the same diseases, but whose targets are quite remote from the myocardial infarction module. And you can see the 4 that we picked on the right. We then turned to our colleagues in the pharmacoepi division, Sebastian Schneeweiss and Rishi Desai, and asked them to look at 2 large data sets they had, the Truven market scan patient database of over 170 million patients, and for validation, the Optum Cliniformatics database, which had over 50 million patients. And we had them ask the following question. Let's see what the frequency is, or the incidence of myocardial infarction in patients started on either of the pairs of drugs listed here, carbamazepine versus levetiracetam, et cetera, over the course of about 2.5 years, and ask the question, is there a difference in that frequency? And looking at these pairs, 2 pairs yielded results that were statistically significant. One was that carbamazepine actually increased the risk of myocardial infarction compared to levetiracetam. And the second was that hydroxychloroquine significantly reduced the risk compared to leflunomide. And as it turns out, just as as our work was being reviewed, there was a paper which highlighted or summarized an ongoing clinical trial that will be used to look at hydroxychloroquine's benefits in non-RA patients and non-lupus patients with respect to secondary prevention. Now, the nice thing about a patient data set of this size, we get real numbers here. So we're talking about 185,000 versus 53,000 myocardial infarction or treatment groups, and that and 54,000 had no prior RA diagnosis in the first case, and 15,000 in the second. So we have significant numbers that are easy to appreciate the statistical power of, given the size of the data set. So based on that observation, this benefit of hydroxychloroquine in particular, we went back to the experimental lab, and Diane Handy and our group looked at potential mechanisms by which hydroxychloroquine could reduce the risk of myocardial infarction. And here what she noted was that hydroxychloroquine's targets, 2 of the known targets, are TLR7 and 9. And she was able to show that by looking at the induction of inflammatory markers, in this case VCAM1 or IL-1 beta, in response to TNF, that hydroxychloroquine suppressed those inflammatory markers as a function of dose. And equally important, TNF-alpha suppresses the expression of endothelial nitric oxide synthase, and hydroxychloroquine offsets that suppression somewhat. So we're not suggesting this is the only mechanism of benefit, but these are at least mechanisms that are consistent with our knowledge of the inflammatory basis for atherothrombosis. Now, as I said earlier, drugs have many targets. This is not just it's not a sort of one-off analysis. In a beautiful bioinformatics study that Chartier published in 2017, I replotted their results, which were given in tabular form. They looked at the frequency of similar binding sites in the human proteome as the binding site to which a drug was developed for a particular target. And as you can see, it's a long-tail distribution that on average, every small molecule drug has 32 other targets, at least theoretically, in the proteome. That's offers a lot of possibility for repurposing or for sorting out adverse effects or the absence of adverse effects. If a drug doesn't bind to any of the proteins that are responsible for prolonging the QT interval, you don't have to do a QT prolongation test, which is an expensive proposition if the theory holds up. So this suggests that really to understand drug effects, you need to understand the pluripotentiality of drug targets in a much more integral way. And to do that, we really need to think first about novel ways for doing a virtual compound screening. Now, this notion of virtual or computational compound screening for drug development really began in the 70s when, with really prehistoric computer systems, the argument was raised that if you had a fast enough system, you could screen a large number of molecules in silico before you ever went to high throughput screening. Now, the chemical space, according to estimates for small molecules, is pretty large, greater than 10 to the 60th. And the computation of pairwise interactions, depending upon what algorithm you use to look for associations, follows to the order of n squared. So it becomes a very computationally intensive process. So much so that if you wanted to screen a huge compound library of, say, 1 million compounds, and you had, in this case, 8,000 targets shown below, which are the number of human proteins whose tertiary structure is currently known, it would take, with the fastest available system now that pharma uses and that we use in the research lab, about 136 years. And if you look at one target versus 1 million compounds, it takes about 150 hours, which sounds right because it takes pharma between 3 and 6 months to screen a single target against their own in-house libraries. So realizing these limitations, we embarked on a project with the Lawrence Livermore Laboratory, and this is in the Department of Energy. This project's funded by the DOE and the AHA. And this project is focused on this notion of accelerating drug discovery. And we use the Lawrence Livermore computational system, which is the fastest or second fastest system in the US. It computes at about a half an exaflop, which is 10 to the 18th computations per second. My neuroscience friends tell me that the human brain goes at about 1 exaflop. Mine may have when I was 12, but I can tell you it doesn't work that way anymore. So here you've got, in our current project, we're focusing on 29,000 human proteins. The experimental structures are imputed for these from orthologs for this great number, and 2 million compounds, all from open access libraries. And our plan is to create a matrix that shows you drug-target interactions in a completely unbiased way from which one could begin to explore potential repurposing strategies. So the last slide is shown here, the last substance slide, and it illustrates this notion of using precision phenotyping, precision reticulotyping, and associated strategies for identifying targeted therapies that are unique to a person's molecular network underlying a specific disease. So we're trying to return from this notion of holism to reductionism back to genomic holism that really integrates this complexity in ways that weren't conceivable previously. And shamelessly, I'll just hawk this book that Barabasi and I and Ed Silverman published about 1.5 years ago that highlights many of the features and theories that I've been discussing today. And I acknowledge my collaborators on the left and the folks who did all the hard work on the right, whose names I've mentioned through the talk. And I thank you for your attention.